the genes that allow some people to be fine just sleeping 4 hours, or we know the genes that allow your muscles to grow bigger. We also understand.
I want both those genes.
Welcome to the Doc Moliver podcast. Plastic surgery, science, and cool stuff in between.
I am thrilled to introduce Enrique Lin Shiao. He is a biomedical scientist and a really nice guy. I met him for the first time a few years ago at a wedding, and I had heard that he had worked in a really special laboratory. I had no idea what laboratory it turned out to be, and that's going to be part of our conversation today. He worked in a lab with a pretty famous Nobel laureate. Let me introduce you a little bit to Enrique. Enrique is a trained biomedical scientist working in the investments department at the Cystic Fibrosis Foundation, identifying, evaluating, and investing in innovative biotech and pharma companies. His education was at the University of Pennsylvania, where he got his PhD in biochemistry and molecular biophysics. He did his master's after he did his bachelor's in Munich. I'll butcher the name in German. He'll tell me later. He's fluent in 3 languages and passable in 2 others. But his degrees were in biophysics and engineering physics. Enrique, you got a very, very unusual background. Where did you grow up, and why become a scientist?
Well, first of all, thanks so much for having me here. I'm very excited to share a little bit about my background and trajectory. So I was born and raised in Costa Rica, but my parents are actually from Taiwan. They left Taiwan in the early 80s, kind of looking for new opportunities. And for me, I always say that they're my largest role models because they left everything they knew behind. They didn't even speak Spanish. They went from Taiwan, which was a country that was very industrialized, to Costa Rica, which at that time was very agricultural. So all the things that they had done before, they both went to college. They couldn't really follow those careers in Costa Rica, and they had to start from zero, where they had a restaurant, they had a business selling clothing from their car. There's so much that they did through that process that I think are part of what really inspire me, and have also kind of informed my decisions as I've moved through the world with my science training. But one of the things that kind of kicked off my interest in science was my dad. He always said the key to success and the key to a better future was through science or through engineering. And that was one of the things that I kept hearing over and over again as I was growing up. So I think from a very early age, that was very almost incepted into my way of thinking. And then growing up in the 90s, I think, was a very exciting time, both in Costa Rica, but also in the world. In Costa Rica, we saw the first astronaut from Latin America go to space through NASA, Franklin Chan Diaz, and he is a born Costa Rican. So being in Costa Rica in the 90s and seeing that someone from such a small country was able to kind of accomplish such a huge dream for us was, it really demonstrated that we could dream big and that we could accomplish big things, even though the circumstances around us weren't necessarily what you would think would make an astronaut. So that for me was super inspiring. And then from the other side, what was happening in the world, the first cloning of Dolly the sheep, for me, that was impressive. It's already a long time ago. I think I was only 8 at that time. But I probably didn't understand DNA or cloning, but seeing that a mammal had been cloned, I think that stayed with me as something very unusual and very exciting. And it was one of the main reasons that I kind of then ended up going into DNA research afterwards. And then I think the very last one that I will mention is HIV, which was huge in the 90s as I was growing up, and thinking about a vaccine and how being able to find a vaccine would be impressive. That was something that was very motivating to me. And we still don't have a vaccine, but we've gone so far already with HIV research and with HIV treatments that I don't know, all of those little things kind of came together to motivate me towards a science career.
It's so interesting how, one little thing, in the news cycle, Dolly was a big thing, but it was very brief, in the grand scheme of things. But it's so interesting to me how specific things imprint on specific people. You were sort of primed and ready to accept something scientific because your parents were so scientifically oriented. So you grew up speaking Spanish and Chinese.
Yes.
Yes.
Spanish, Chinese, Taiwanese, which is a dialect in Taiwan, a mix of all those. So even now when I'm speaking to my family, we have a mix, or we take words from each of those. And it's kind of a family language at this point where if you don't speak all those three, it's hard for you to follow what we're saying.
It's your own scientific code. How many languages do you speak fluently?
So now I speak 3 languages fluently: Spanish, English, German, and then I speak French, Mandarin, Chinese, and Taiwanese, not as fluently, and with a very strong Costa Rican accent as well.
That's fantastic. So in your training, how did you wind up, having international parents and you being a first generation Costa Rican, but parents who just came over as adults from Taiwan? I guess you must have had some sort of flavor for why not go someplace else? We don't have to stay here. I know Costa Rica is your home. You were born there, but how did you wind up going to Germany for your training? Here in the United States, that's sort of the extreme elite get identified and said, you need to go to London School of Economics, you need to go to et cetera, et cetera. Maybe people go there for a semester. But I think in other parts of the world, it's much more common. What was your pathway?
That's a great question. I'd say one of the things my parents wanted when they left Taiwan was actually to come to the US, but they just didn't have the funding. They didn't have the support from their family. But they always viewed Costa Rica as kind of a transition point, where another thing that they kept saying when we were growing up is, you can't stay in Costa Rica. We came here so you get better opportunities. As we were growing up, they would always say, okay, you have to push hard with your academics, you have to do really well, and you have to leave when you turn 18. You can't stay in Costa Rica. So they pushed us a lot. But the other thing that they did was they tried to put us in the best schools that they could. So they did a lot of sacrifices throughout our childhood, where they wouldn't go out to dinner, or they would save as much as they could to pay for the best education. And in their minds, the best education was the German school in Costa Rica, which provided a trilingual experience. And also it wasn't as expensive as American schools in Costa Rica. Me and my 2 brothers went through the testing that allowed us to go into that school. And then what ended up happening is, as we finished, my brothers are older than me, but as they finished and went to college in Germany, my parents realized that there's no tuition in Germany. So they were so relieved that they saved so much money by sending us to Germany instead of the US.
What a lucky break.
Yeah, so that was kind of how we all ended up going to Germany. And then, my parents pushed me throughout my high school. I graduated top of my class, got a scholarship that allowed me to pay for my living expenses once I went to Germany.
So you went to Germany for college, not for high school?
No, I went to Germany for college. And people are always ask, why is there a German high school in Costa Rica, and think that's super random. And the reason is the German government believes that if there are Germans outside of Germany, they should have access to German education. So a lot of the top executives in companies like Mercedes-Benz, BMW, those are people who are sent from Germany, and their children have to have access to a German education. So almost in every single country in the world, there is a German high school where those children can go to, and they've opened it up to people from that country as well.
That's fascinating. Well, let's skip on, because we've got an hour and we've got a lot to fill up. We've all heard about genetics. Tell us what is epigenetics?
Yeah, so epigenetics is really what I focused on during my time I spent a year doing research in Cambridge in the UK, and then almost 6 years doing my PhD at the University of Pennsylvania, really focused solely on epigenetics. And for me, it's a fascinating area where if you think about human beings or really any organism out there, we start from a single cell and that cell creates a whole human. And for me, that question of how that single cell can create a lot of different types of cells like teeth, eyes, hair, skin was super fascinating, especially because if you think about each of those cells, they all have the same DNA. Each of those cells have the same instructions. So what determines whether that cell is an eye cell or a tooth cell? And then a lot of it comes to how it's organized within, how the DNA is organized within the nucleus of that cell. If we imagine DNA as an encyclopedia, then in some cells, only some chapters are being read, while in others, other chapters are read. For example, in the eye chapter is going to be read, while the rest of the DNA is going to be packaged really closely, and those chapters are never going to open. So epigenetics really is the study of how DNA is organized and how that influences things like development of a human being or a living being, but also how it influences disease. For example, if we think about cancer and cells in our skin, skin cells shouldn't be able to live when they're not stuck to each other. But in some cancer processes, skin cells learn how to live independently. They can go into your bloodstream, they can travel to other places and take cancer to other places where they shouldn't. And basically, they're reading chapters that they shouldn't be reading in that encyclopedia analogy. And for me, that was part of what I was studying. I was trying to understand how people became resistant to certain cancer treatments. And a lot of it was connected to how DNA was organized within those cancer cells. And that was fascinating to me.
So let me ask you something on a very simplistic level. When we talk about stem cells, you know, we play around with stem cells and put them in different environments next to different chapters, and that seems to, you know, guide them in terms of their development. Is that sort of similar to what you're talking about?
It is similar, yeah. People are very excited about stem cells because they theoretically can allow us to create all these tissues and different things that we might be missing. For example, if you could, instead of needing a dental implant, which I'm currently waiting for one, you could just inject stem cells there and grow a new tooth.
That was in the news lately about doing exactly that, wasn't it?
True.
Yeah.
Very recent. Mm-hmm. So there's so much that's connected to those stem cells and how we can push them in one direction or another by making them read certain chapters instead of us.
So it's exactly the same as what you're talking about.
Basically, yeah.
Yeah, that's very interesting. Okay, so we're heading into genetics. Let's get to the meat and potatoes of it. What is genome editing? And I'm going to expect you to talk about CRISPR and Cas9 and origin and potential. I'm just going to let you instruct us and teach us for a little while.
Yeah, I guess just starting from the beginning, genome editing is a way that we can go into the DNA and kind of change certain bases. And if we think about diseases, there's a lot of genetic diseases out there, and they really are primarily the reason we have them is because there's changes in our DNA that have accumulated that can lead to certain diseases because they change the function of genes, for example. And if there was a way to edit and go in and kind of switch that back to the normal sequence, then we could get rid, in theory, of those diseases. In practice, this is very complex and it hasn't happened, or it's a field that has been growing for decades very slowly until I would say about 2012, when the field completely transformed. And that is thanks to the technology called CRISPR-Cas9. And if you haven't heard about this technology, it's amazing. And for me, it's fascinating what you can do with it. And it's already changing the world. But it really stems from scientists who are looking at how bacteria defend themselves from different threats. And bacteria, like humans, can be infected by viruses. And one way that bacteria can protect themselves from viruses is by using this system called CRISPR-Cas9. CRISPR-Cas9 is a way for bacteria to kind of store pieces of DNA from those viruses that have infected them in the past into a library and then they can access that library and compare every time they get an infection. Are these sequences from a virus that infected me in the past? And if so, I'm gonna go and chop that virus into pieces. So it's a way of kind of cutting DNA. And if you think about editing, if you're thinking like a podcast or a movie, the first thing that you have to be able to do is to cut, and then you can start pasting and moving things around. But if you can't cut in a precise way, that is really the hardest part, I think, to start editing. So this technology in 2012, two people, Jennifer Doudna, my former boss, as well as Emmanuel Charpentier, were able to take out that system from bacteria, reprogram it, and realize how they can really make it go to almost any sequence of DNA and cut in that precise place. And that sounds very simple.
No, it doesn't. No, it doesn't. No, it doesn't at all. It sounds unbelievably hard when you think about how long a DNA molecule unraveled is, and to send it in and cut something exactly between this base pair and that base pair. It's insane that anybody could figure that out.
Yeah, and to maybe harp a little bit on that, if we think about how long the DNA in each of our cells are, we have 3 billion letters of DNA. If you were to take 3 billion letters, paste it into a Word document, you would cover 1 million pages. And then, well, just picture or try to imagine 1 million pages of Word full of letters. CRISPR has the capability to go into those 1 million letters and find a stretch of 20 letters, almost anywhere in that 1 million pages, which is insane to me that it can go there and then just cut in that very precise place. So that's really why they got the Nobel Prize in 2020, because this technology opens up so many possibilities.
That's some fantastic stuff. When I read the book, Code Breaker, that was the name of it, right?
Yes.
Yeah, Code Breaker, which is sort of a biography of Jennifer Doudna by Walter Isaacson. I was simply blown away because of the possibilities. And when I told my friends about it, I actually was reading about it as part of a book club. I read this book, and I said to the book club, we need to read this. And we wound up reading it, and we had you by Zoom that evening. That was our couples night, and you so eloquently described it in a much shorter time period. But what an amazing read. What was remarkable to me after reading that book was that in science, in medicine, you read an article and they say, oh, in mice, we just did this, and hopefully in 20 or 30 years, this might lead to something. And my impression from reading that book was it's not only going to lead to something, it's by the time the book got published, it's already doing things. There's labs all over the world doing stuff in very small settings with not a massive amount of equipment, which brings up all kinds of scary prospects, but it also brings up sort of like open source computing. I mean, it's like it's everybody's playbox, everybody's sandbox. So can you talk about that a little bit?
There's so much to cover here. I think one of the things that I was thinking about as you were saying this was this technology only came out in 2012, but already over 200 people have received CRISPR treatments. So it's already being used in humans. And one example that is often cited is Victoria Gray. In 2019, she received a treatment for sickle cell disease, disease where...
that was in the book, yeah. Right, and it was $1 million shot, and of course Bill Gates is supporting this heavily with funds, and they're scaling it up. So it's amazing.
Yeah, and this patient before this treatment, she was bedridden. She had to go to the hospital several times a year because she had all these clots and all these issues with pain. And now, since that treatment, she hasn't had to go back for any supportive treatment anymore. So it's not only amazing, it also saves a lot of money when you're thinking about all these supportive treatments that these patients needed before. Now she doesn't need. She's able to travel. For the first time, she went to outside of the US, I think, this year. And to me, it's amazing what you can do with this technology. And at the same time, we have a lot of other clinical trials out there for blindness, for some other blood disorders, liver disorders. The possibilities are really exciting here. I think the other point that you kind of hit a little bit on is there's also the potential of some negative outcomes, or there's a threat here as well, because if we can edit the genome, there's so much that we can edit. And there's a concern here where people think a little bit about what should we edit and what shouldn't we. For example, we do know the genes that allow some people to be fine just sleeping 4 hours, or we know the genes that allow your muscles to grow bigger. We also understand.
I want both those genes.
Yeah.
Go ahead.
Or we also understand genes that, there are so many things that we already, or for example, pain. We understand which genes can contribute to pain and which ones, if you could tinker with them a little bit, you could change chronic pain. But if someone wanted to go rogue completely and then combine all those three things, you could think about a person who doesn't have to sleep much, doesn't have the same sensitivity to pain, has increased muscle mass. Are these things that we necessarily should be doing or not? Is this enhancement versus treatment? There's so many questions here around ethics that are a little bit hard to tackle, and I think we need society to come around and discuss openly.
Right, and the scientists are around the world attacking, or to use a better word, they're combining, they're talking together to try and limit what can be done and what can't be done. And most of the countries are signing on to govern this and regulate it, aren't they?
Absolutely, yeah. Most countries right now, if not all, forbid human embryo editing for reproductive purposes. Even in China, human embryo editing is for reproductive purposes is prohibited, as was in the US. That being said, and you mentioned this briefly before, there was a case in 2018 where a Chinese researcher, He Changkui, he did embryo editing, and 3 babies have been born since that have embryo editing, which was a huge concern. This person went to jail for about 3 years. He has since left jail and now has his own research lab again, which is also concerning. But
and in that case, he made them all resistant to HIV, didn't he?
Yes, that was the plan. It seems like it might have worked in one of the babies, but it's unclear if it did work in the other 2.
Okay.
And then there's questions about what other consequences does it have to kind of tinker with that receptor that doesn't allow HIV to come in, but it also kind of leads to other worse outcomes with other diseases.
Unintended consequences.
Yes. And it's hard to really justify when HIV is a disease that you can use treatments at this point. And you can also prevent.
Right. You can take 1 pill a day and live a totally normal life. Let me ask you about delving into curing someone of a disease. For example, the lady in Chicago who got cured of sickle cell, and curing it for her prodigy. So there's a big debate or a dilemma about that. I can cure you of disease XYZ for just you during your lifetime, or if I catch you early enough in life, I can make it so it's cured in all of your sperm or all of your eggs, so that you can't pass it on anymore. Is that true?
It could be true. I would say right now it's not allowed. And there's questions here of, is it necessary?
Right.
For what diseases is it necessary? Oftentimes, you can be a carrier for a disease, and there really isn't a necessity then to do a treatment. Or if you're going to go to the point of editing a gamete, a sperm or an oocyte, why not then just select the ones that don't have the defective gene? And then the other aspect here is we don't really have the technology, or it's not as advanced that we can go in and edit sperm or oocytes safely right now, or really, it's not really feasible yet. We could get to that point, that's one day, but we're not there yet. And then the other question is, people have talked a little bit about embryo editing, but as you know, the embryo starts to divide relatively quickly. So if you're trying to edit an embryo, what could happen is that some cells get edited and some others don't. So you end up with this mosaic where maybe you have disease in some cells and not in others. And what does that mean long term for that person?
Right, right. Let's move on. What are you working on now? What do you do? What do you do with your life now that you're not a scientist, or you are a scientist, but you're not in the spotlight of Jennifer Doudna's lab? So what are you doing now?
Yeah, right now I'm working at the Cystic Fibrosis Foundation, and there are current treatments for cystic fibrosis that treat about 90% of patients, and it's really kind of a miracle. Small molecule drug called Trikafta, and it's really amazing. But for that last 10% of patients, the mutations that they have are not treatable with this small molecule. So really what we're excited about thinking about is how can we go in and fix that gene using, for example, CRISPR or gene editing technologies. So a lot of the time I spend looking at the most innovative startups that are working in this space, trying to see if their technology is suitable for correcting this defective gene. There are other challenges here because cystic fibrosis is primarily a disease in the lungs. We don't necessarily have a way to deliver things to the lungs or genetic medicines there yet, which is also why if you look out at CRISPR companies out there, most of them are working in blood, liver, eye, tissues that are already accessible with the delivery vehicles that we have today. So for me, if we're able to get to a cure for cystic fibrosis, it really opens up the possibility of curing so many other diseases, brain diseases, other organ diseases, and CFF, or the Cystic Fibrosis Foundation, is also an amazing place. Thanks to the investments that they did in the past for this drug that now treats 90% of patients, we've been able to get back $1 billion in royalties to the foundation that we now can invest in this new, very innovative technologies. So it's really an exciting space to be in, and it's very rewarding as well.
So I think that's a misunderstanding by a lot of people. A lot of people, myself included, thinks, oh, you're going to use CRISPR-Cas9 or to treat cystic fibrosis. So what does that come, a little syringe, and you jab it in your thigh and you're cured? And of course, that's apparently not the case, and neither is it the case for a whole host of other diseases. It's really comes down to delivery and where is it going. There's things like the blood-brain barrier where you can deliver it peripherally, but it doesn't really get up to the brain for various diseases. So that's was eye-opening to me. You just can't shoot someone in the leg and make them real muscular, or maybe it'll make muscles right in the leg there, but so it's there's a lot of complex issues here, and delivery and disbursement and what other things are going to be attacking it and defending against it and what have you.
And I think one other one that people are not necessarily aware of is CRISPR, because of its bacterial origin, once you inject it into a human body, you're going to get an immune reaction. So this means very likely you're only going to be able to receive one single treatment of this, and it has to work efficiently that one time. If you try to redose, your immune system is going to attack that from the beginning. You won't get any further editing. This is what most of the field thinks right now. There might be ways to kind of get around this, but we don't have a solution as of yet. So it has to work really well, and it has to deliver very well. And I think those are things that I think a lot in my day-to-day. I'm looking at delivery companies. How efficient is it? Is it going to the cells that we want? Is it going to those stem cells that are going to last for a long time? And I spend a lot of my time thinking about that. And then from the other side, CRISPR was just the beginning, being able to cut. If you want to insert and do the paste function, there's so much that you have to overcome there as well. How do you deliver the thing you want to paste? How do you paste the entirety of it and not just pieces of it? So there's a lot of technology questions here that haven't been answered, and that make it more complicated for diseases where you need to replace the gene and not just cut out something that's defective.
Let's skip over for just a little bit. It's very controversial, but I'm going to get a vaccine for COVID, you know, I don't want to die, and I don't want to drop dead in 2 years, and I don't want to do this, and unintended consequences. A lot of misinformation out there. You know, sometimes in medicine, we're looking for a 99% bullet. And we know that there's a 1 or 2% chance of these untoward effects. And that happens in vaccines all the time. And a lot of people don't want to hear that, right? We can save the vast majority of people. We can prevent the vast majority of people, but we have to accept a little bit of downside. And people, when they hear that, when the downside is you or someone you love, it's heartbreaking and you can't deal with it. But nothing really is 100%. I'm probably talking about two different things here, but can you talk about this a little bit?
Yeah, no, I'd say there's definitely a lot of misinformation out there, and the amount of information is almost overwhelming. So for people who are looking at this, it might be hard to kind of go into the weeds and understand the numbers. When we look at mRNA therapies, they are extremely safe. So it's not only 99, it's like 99.5 or even higher. There's still going to be a risk. I think there has to be a cost-benefit analysis as what is the higher risk, the one from this treatment or the one that I would get if I get sick with the disease that I'm trying to prevent. And in most cases, it's the later, where there's going to be a higher risk if you get the disease. So as a society, we think it's better to prevent that. And then the policies kind of go in that direction where they recommend vaccination, for example.
Right. One of my friends, when the COVID pandemic broke out, he said presciently, we really need to just vaccinate old people and quarantine them and let everyone else loose, sort of like Sweden did. Turned out to be probably the right way, and a lot of people and a lot of states are now adopting this kind of way of doing things. I think the government is endorsing that now. Do you have any thoughts on that?
I think we are in a point now where most or a vast majority of the people have either gotten vaccinated or have been exposed to COVID. So there is some degree of immunity out there, which is why I think a lot of governments are endorsing this now. If we go back to 2020, I think the information was just not enough to make the decisions. So to me, it makes sense that when the information is not enough, you go for the more careful approach,
right,
and you try to be more careful and prevent other issues. During the pandemic, I was working in a COVID diagnosing lab, and I remember when we're trying to verify that our diagnostics was working, we got samples of COVID, and this is maybe April of 2020. And my hands were shaking and I was wearing all this PPE because we had no understanding of the disease. And I needed to show that our diagnostic test could effectively work. So I needed to do it with positive samples. And I remember that fear. And I mean, at this point, we've been living with this virus for so long that I wouldn't feel the same way as I did in 2020.
Right.
So I can see how policies have changed since.
Sure,
it makes
sure.
sense. One thing that I did want to mention is we talk a lot about human health, and that's probably because it's our interest overall. But CRISPR, because it's editing DNA, it can really edit a lot more than human cells. It can edit agricultural products, for example, and it's already been used to edit over 280 organisms in the world, which is really exciting. And I think one of the points that I don't talk too much about, but I think is really interesting, is in the agricultural field, where you could think about all these different, like tomatoes, corn, all of these different products can be affected by disease as well. And we can use CRISPR to prevent a lot of those. Or as the temperatures are rising, I don't know, this summer was so hot, at least up here in Austin, I'm sure in Houston as well. But as the temperatures rise, like this summer, then a lot of products might not survive that heat. But we can create products using CRISPR, like tomatoes or others, that can retain water more efficiently and then be able to survive in a lot drier conditions. Or if we think about deserts, we can start to grow corn in the desert, for example, by using CRISPR to really change the properties of those plants. And for me, that's also a whole other possibility that is far from what I do, but is just as exciting when it comes to CRISPR.
That sounds really fascinating. You worked with chemistry Nobel Prize winner in 2020 for genome editing, Jennifer Doudna. First, did you have an opportunity to meet or talk with Walter Isaacson? And two, beyond that, what was that whole experience like?
I did, yeah. So I started in her lab in 2019. Walter Isaacson was coming in and out of the lab. I was there when he was doing his first CRISPR experiments, trying to understand the process. But yeah, no, it was an amazing experience. I remember going to my interview with Jennifer Doudna and being so starstruck and nervous, even though she hadn't won the Nobel Prize yet.
Was it a given? Was it a given at that point? Did everyone know she's going to get it?
People pretty much knew because CRISPR had grown so fast and it was being used in all over the world.
Right.
And the potential was already there, that we knew she was going to get it. So I remember going to that interview and almost shaking before I met her.
Right.
I was so excited, but she is just such a nice person as well. And her vision and the way she does science is also very exciting. So it was really a great experience. I was there, I think mid-August is when I started in 2019. And then the pandemic hit, unfortunately, at the beginning of 2020. So a lot of my work, which was kind of focused on that paste function of CRISPR, where how do we replace and put bigger things back into the genome, a lot of that had to pause while we were working through the CRISPR diagnostics, the COVID diagnostics, and waiting for the pandemic to kind of unravel a little bit. For 6 months, the entire lab was closed. And California was very restrictive. Even places like the dog park, which was outdoors, was closed.
Wow.
Which might have been a bit excessive.
might have been. I still laugh when I see people walking down the street in a blowing wind with their mask on by themselves.
Yeah, that's true. I would say we were in California. Even hiking outdoors, people were wearing their mask.
I know. It was kind of funny. Anyway.
So, yeah, no, but I was there when the Nobel Prize was announced, and the announcement happens in Europe. So it was, I think, 3 a.m. at night. And people, every year there was the same rumor, Jennifer Doudna is going to get the Nobel Prize this year. Usually I kind of ignored it, but that year, since I had just joined the lab, I was like, oh, I'll just, I think I woke up around 2 a.m. and I was like, oh, I might just check it out and see if something's happening. And then it turned out that she got it. We all went to the lab to celebrate. It was still, I think, October of 2020, so everyone was masked. Everyone was trying to stay kind of far from each other, but it was still a huge moment and celebration, and very exciting time to be there in the lab as well.
I understand she's a very nice person, and she's all about open. You know, in the book, there was all this information about patents, and of course, you know, in science and industry, patents are just like the bane of the scientist's existence. But it's where the big money comes from, and the big money goes to. I don't want to go on too much about that, but it was a tense. There was a tense couple of years between the East Coast and the West Coast patent fights and the law stuff, and I'm sure that was difficult for everybody.
Yeah, and I think especially for patent lawyers, it's probably fascinating as a story. I think for the scientist itself, we kind of try to stay away from letting that influence our day-to-day. But I think it's really interesting. A lot of the discussion was around who had invented what part of CRISPR, genome editing. Right. Jennifer Doudna and Emmanuel Charpentier had really demonstrated in their 2012 paper that you could edit cells, and that this technology could edit or be programmed to edit almost any sequence. But they didn't go all the way to show that you could edit mammalian cells, for example. Right. So that really was where the question lay, where if you understand the technology, that step going from programming to editing mammalian cells is relatively straightforward. But what the patent dispute is in how straightforward is it? How do we define straightforward? Was there inventive steps on top of that? So it starts to get very jargony in terms of lawyer.
Right. Very
things.
sticky. Very
Yeah.
sticky. All right, let's change the topic. Given the recent pandemic, and we touched on this stuff already, are there ways we can use genome editing to protect ourselves from infectious disease? And we'll go back to the book. In the end of the book, it's sort of like a cliffhanger. They talked about the scary side. And so let's. I hate to even put this out there because people will listen to this and they'll say, "Oh my God, whatever." But there are some nefarious people out there who could take this science and take viruses that are not good viruses, dangerous viruses, and do things with them. So is that really true? Can that happen?
Yeah. Let me think. Where can I start?
Let's go back. Let's go back to the first question.
Yeah.
Are there ways to use genome editing to protect ourselves from infectious disease?
Yeah, and I think this is a great question. And this was something that scientists also debated from the beginning, because this is how bacteria protect themselves from viruses. Can humans also use the technology in that same way? And it turns out that we can. There are companies out there. I think Excision Therapeutics is one that is trying to use CRISPR to cut out HIV from human cells. So HIV is one of those viruses that integrates into your DNA sequence. So because CRISPR allows cutting, if you take it and cut the two ends where it has gone in, you could, in theory, remove HIV from those cells, and then in that way cure the disease. So there are people thinking about that. There are people thinking about other infectious diseases. Could you kind of seed bacteria or something to protect you from infectious diseases in the respiratory tract? So there's a lot of...
So you put bacteria into the body that we live in harmony with. Look, our biome is full of billions of bacteria, which is a fascinating topic all by itself. And those bacteria are your little grunts. They're your little army men and women, army persons that are out there fighting the virus because they know how to fight the virus because they've been doing it for eons.
Basically, yeah, there are a couple companies out there. I think Sniper Bio is one of them. They're also thinking about, can we use bacteria in your microbiome to maybe deliver enzymes that a patient might be missing, for example, in PKU and some other diseases that are more metabolic and enzymatic. So there's so much potential still, not just by editing the human cells directly, but by using these other organisms that live inside us.
I read about a company in Australia that takes people with congenital hypercholesterolemia and are adjusting. Did you hear about that?
Yes.
Yeah, and apparently there's actually human studies going on right now where they get the treatment, and these people that run cholesterol of 500 and 600 and 700, who literally get vascular disease when they're 25 and heart attacks when they're 30, these are the type of people we're talking about. But they can treat them, and without further ongoing daily medicines that really don't work on these people, they can lower their cholesterol levels.
Yeah, no, I think that's fascinating. And it's actually one of the most common indications that gene editing companies are thinking about, because it's so well understood. There's this gene, PCSK9, that is connected to hypercholesterolemia. And what I think a lot of these companies are trying to do is just going in and disrupting that gene in the liver. And apparently that can reduce the levels of cholesterol so dramatically that a lot of companies are trying to work in this space. So it's actually pretty exciting to see what's going to happen.
Yeah, if you got this disease and you're going to be, having a stroke by the time you're 28, I would expect that there's going to be some countries and some states that'll say, okay, we're giving you the go-ahead. Go ahead and try it. And people will say, I've got nothing to lose. Of course, it's going to be pretty safe, and they'll have looked at, animals and whatever, but that sounds fascinating. Let's go back for
yeah, it could be out there in 5 to 10 years even.
Yeah. Let's go back to the last little blurb, because you have to do this in a podcast. Let's do the cliffhanger. So can people make – so two things. One, from what I understood from the book, again, people can make really bad viruses. But the good news is, because of the same technology, at least our government and our military and DARPA is looking at test kits that can spot like 100 viruses just by a little drop of blood. Is that really true?
I wouldn't say just by a little drop of blood, because
Okay, I'm embellishing. Go ahead.
that goes a little bit too close to Theranos and some of that controversy.
And she's in jail.
Yes. But I would say that we are developing better and better surveillance systems and diagnostic systems that are more sensitive. I don't think a single drop of blood would allow it, but maybe in the future, not a single drop, but a little bit more than that. I think at the same time, as we think about CRISPR, there are some concerns that it can find certain sequences, and then you could tailor it towards certain people, for example, who all share that sequence. And there's some concern around there, but there's a lot of investment from the DoD as well as other entities looking at how we can use anti-CRISPRs to kind of stop, for example, a potential CRISPR attack. And that's also a fascinating space where anti-CRISPRs are things that have developed by the viruses that attack the bacteria so that they can stop the CRISPR system from killing those viruses in that bacteria. We've been able to kind of isolate that as well. And now we can use it to modulate CRISPR as in other organisms, which I think is fascinating. But there's a huge investment as well from our like the DoD, just in case someone's thinking about going rogue and using these technologies for something they shouldn't.
So we're working on it. We're ahead of the curve, hopefully.
Yes.
Yeah. Well, I think we're just about time to wrap it up. This has been a small part of what probably could be like a 2 or 3 hour conversation.
Absolutely.
Thank you so much. I think people will find this super fascinating. I do. I'm kind of nerdy that way. All right. Well, that's it. Thank you so much. I appreciate it.
Yeah, no, thanks so much, Clay.
Thanks for visiting. See you next time.